speaker today is Sarah Santos. She's a PhD candidate and graduate Assoc Fellow at the School of Engineering at Brown University. She's doing interdisciplinary work in fluid mechanics, biology, and ecology. She holds a master's in aerospace engineering from Iowa State University and a bachelor's in mechanical engineering from the University of Akron. Her work focuses on understanding the fluid structure interactions between swimming organisms and their environment using robotic platforms and live organism experiments to characterize swimmers' kinematics, morphology, and hydrodynamics. Her current work looks at metachronal swimmers, such as shrimp, to design bio-inspired robots to explore marine environments both on Earth and moons throughout the solar system. So please join me in welcoming Sarah.
Thank you so much. I will start off by saying if you have questions, just raise your hand, please. Do not make me talk uninterrupted for however long. I would really welcome any participation that you have. Also, I've been doing this work for the past 5 years. So for me, I think everyone knows what I'm talking about because, well, why wouldn't they? You know, it's so interesting. But if you don't, please ask questions. The presentation will build up on a lot of the things that I'm going to speak of in the first few slides. So if you're lost, just let me know and we can all move together instead of leaving people behind as I go along. Yeah? Thank you, Savannah, for the introduction. My name is Sarah. I work with Professor Monica Wilhelmus at Brown, and I am co-advised by Dr. Neil Stack. And I'm going to be talking about shrimp as a model organism for bio-inspired underwater vehicles. Here you can see a video of shrimp. This is right field PIV. This is a Hawaiian shrimp. It's about 1 centimeter and it swims metachronally. I'm going to talk about that. But first I kind of want to contextualize my work, let you know what it is you're going to see. So I'm going to spoil the whole talk for you, hopefully, if I've done this correctly. So what we do is we do 2 things at the same time. We do biology-inspired engineering, and we do engineering-enabled biology. Both of these come as the result of the same set of experiments. So we look to understand the fundamental principles of shrimp locomotion to then understand the ecology and inspire our engineering. So for the engineering side, we're looking to create more maneuverable, task-specific underwater robots. For the biology side, we're looking to understand this propulsion mechanism because it is present across the intermediate Reynolds number regime, and a lot of the animals in that regime swim using metachronal swimming. So we think there might be some sort of evolutionary pressure that has led these animals to swim in this regime. So we're looking to answer 2 questions at the same time and hopefully only do the work once. And here, I do have a pointer digital one. So this is what the appendages of shrimp look like from the frontal view, if you're looking at a shrimp in the face. And they have 5 sets of appendages, 2 on each side. They have a solid membrane, and then surrounding the solid membrane are bristles or hairs. And then they have this little appendix here that connects both of them so they don't get too spread out. And we're looking at this first, like I said, from an engineering perspective and developing robots for underwater locomotion. So I think the crowd is the perfect crowd for this. You both, and hope, are familiar with ROVs and AUVs, and they are excellent instruments, but they have their caveats. ROVs are tethered to a ship, have low maneuverability, and AUVs can do very fast swimming, but have limited maneuverability. What we're looking to find is this intermediate or in-between region where we can have a metachronal swimming enabled robot that can go in between and can fulfill the functions that are not fulfilled by those 2 types. For example, if you want to navigate a kelp forest with your AUV, that is going to be extremely difficult. Can we engineer a robot that is small enough, maneuverable enough, that can navigate those complex environments without creating damage to the environment. So I have the same shrimp here. And people have started to look at this. I think recently, I would say in the last decade, people have really started to think about inspiring robots in this propulsion mechanism. So the first, I think, publicized robot on this was the Krillbot. This is work from a team at Oklahoma State. And they inspired their robot in krill. And they have this robot, 5 legs, and it swims metachronally. So this was the first attempt, which is a wonderful attempt, and it allows us to explore a parameter space. For example, how far can your appendages be, or how far do they need to be for you to get maximum efficiency? Should they all be beating, with the same phase lag? Should they be quicker? So this allowed us to explore that parameter space, but it really lacked the complex morphological and kinematic details that make shrimp such an interesting swimming organism. So what we're doing, and I'm going to show you this is kind of a clickbait image is what it is. This is our Tenure project. So this is our goal for the next 10 years, is to create a robot that looks like shrimp. The antennae can sense, it moves metachronally. It has a quick escape mechanism by using the tail, and it can do all sorts of mapping and sensing. Right now, we're not there yet, but we are on our way, and I'll kind of go through the process of how we started that and where we're at right now. So why shrimp? Shrimp are the most amazing model organism, and I am extremely biased, but I will disagree with anyone who says otherwise. They do a lot of different things. They use their antennae for sensing, which they can move individually to sense the flow around them. They use their tail as an escape mechanism, so if you scare a shrimp, it will jump so quickly. And then they have a compound eye, so their vision is very different from what we see. And their propulsors are very interesting, and that's what I'm going to focus on today. I will focus on their propulsors, and the truth is my PhD work has been a rabbit hole of looking at propulsors and going into more detail and more detail and more detail until I've explored the most infinite detail. And thankfully, someone else came along that took over the large picture work otherwise. So Metachronal swimming is the name given to the wave of appendages. So shrimp start by beating their very last appendage first. And then a little bit later, which is usually 25% of the amount of time it takes one appendage to do a full cycle, so a power and recovery stroke, the next one will begin. So this is P4, P3, P2, P1 is the very last one to start its beating cycle. And one thing that is interesting is you can see here, when they return together, but when they do their power stroke, they do their power stroke individually. We have found, and this is not my work, so I won't speak too much about it, but we have found that this helps them reduce the amount of drag during their recovery stroke. Because I'm focusing mostly on the appendages, when I talk about Reynolds number, I'm talking about Reynolds number in the intermediate regime. This is from 1 to 1,000 in my case. I know intermediate means very different things to very different people. But mine is from 1 to 1,000, and shrimp appendages or pleopods, they are in the range of 1,000, whereas their body is around 3,000. And I'll talk about that, and this is the formula for the oscillating pleopod, which has the frequency, the amplitude, and the length. So this is from the side view. We see what shrimp are doing. They are moving their appendages with a phase lag. Now let's look at what they're doing from the back. This is where it gets even more interesting. So you're seeing a shrimp from the back, and you can see during their power stroke, their appendages spread out completely. This is because they use a drag-based thrust to propel themselves forward, and the bigger the area of those appendages, the more thrust it will create. And they have, like I showed you before, they have the membrane part and they have the hair part. And one of the things I find funny is that there's a little bit of redundancy in shrimp, so you can lose a few hairs and still be okay. As you can see from this shrimp, it's lost a little few things, but it's still going. And then what they do during the return stroke is they tuck in all of their appendages. So all of these hairs, all of these membranes fold together. And through this, through creating more thrust than drag during their recovery stroke, they create net thrust. And this is how they move. Now there's a lot of details that we could explore here, and I will talk about a few. If you're interested in the rest of the details, please feel free to ask or email me. I'm always happy to talk about shrimp. Happier than I should be, to be honest. So I will go into my first project by talking about biology-inspired engineering. And this is one appendage of shrimp, and is an appendage that we designed and printed and validated. And I'll go through how we got there, what we are including here, and I'll go through some results. Before I move on, does anyone have any questions? Okay, crickets. We'll move on then. So first, to really have a system that is a true representation of the animal that you're looking at, you need to understand the animal, how it moves, what it's doing, and how much complexity you need to add to your system to have good fidelity between your robot and the animal. One of the most important things for shrimp is the kinematics of their appendages. So there are a few angles that rule these kinematics. I'll go through them. I'm going to be talking about these angles throughout the whole talk. So if you in slide 10, need a reminder of what they are, please let me know. I confuse myself sometimes. It's just, there are a lot of subtleties here, and I really want to make sure that you can appreciate them as much as I do. So shrimp have four main angles by which their motion is characterized. The first angle is alpha, right here. That's the angle between the protopodite, which is the top part of your appendage, and the body axis. So here in our robot, we see alpha right here. This is the top part. Then beta is the angle between this top part and this bottom part. The bottom part is a biramus appendage that includes both the endopodite, which is inside, and the exopodite, which extends out. What we did in our robot is that we consider these to be two extremely important parameters, and we wanted to control them as finely as we could. So we made these two mechanisms active, which means there's two servos controlling these two angles, alpha and beta. And as a validation of our robot, I'm showing you the kinematics curves that are averaged over 5 cycles. And in the black dashed lines, you see data for krill. This data is from Murphy. He published a really nice paper in 2011 describing all the kinematics of krill, and that will become very important throughout this talk. But we have the kinematics. There is a shaded gray region that is the standard deviation, is very small, and I'm very proud of that. Next, there's this angle that is so important. This is the gamma angle. So here you have your endopodite, and here you have your exopodite. This angle changes throughout all of the cycle of shrimp swimming. During the power stroke, this angle becomes greater. During the return stroke, this angle becomes much smaller. we wanted to have this actively actuated, but was extremely challenging to get this out of plane motion. And as it turns out, you don't really need to have that out of plane motion to be true to the real animal. And so this is passive, and it is actuated by the fluid structure interaction on this paddle. These are the kinematics. There is no comparative data for krill. This is difficult to measure. And so I am only plotting the robot there. But as you can see, even being actuated by the fluid structure interactions, the standard deviation is pretty small when you compare with the amplitude of the whole stroke. And then the third image shows this fourth angle. This angle is the angle zeta. It had not been characterized before. No one really had paid a ton of attention to it, but because I looked at so many shrimp pictures, I was like, maybe we should include this. And I was very obsessed with it, and we decided to include it. So this angle is a little bit difficult to explain, but if you have your endopodite here and you have your exopodite here, they're not both going in the same plane. Your exopodite is actually at an angle, so it's going at an angle. This is also what allows the exopodite to be passive with the gamma, because this little offset angle allows the fluid to create side forces that open and close the exopodite. So this is fixed in the design, so it never changes. It continually the same throughout the cycle, and we really haven't characterized this for shrimp. It may or may not change throughout the power and return stroke, but we're not sure yet. So for now, we're going to consider it to be fixed. This is what our robot appendage looks like. Here you have the two servos that actuate it. You have a gear train that actuates this first part. And then you have a second gear train here that continues inside of this first part that actuates this appendage here. So both of these are controlled by us, both of these angles. And here you see the side view. So this would be the protopodite. This would be the conjunction of the endopodite and the exopodite. And here from the front view, you see the endopodite here and the exopodite. And as you can see, during the power stroke, which it's doing right now, it opens, and during the return stroke, it tucks in, and both the endopodite and exopodite return as one. If you are interested in 3D printing this, it's all right here. It's on Zenodo. It's 98% 3D printable. You just have to buy the servos yourself. And we have the MATLAB code that prescribes the kinematics. So all you need to do is 3D print, use MATLAB and an Arduino to prescribe the kinematics, and you can make your own little paddle. My goal was to make an army of these. We may come up short, but it's still a good effort. I'll have this QR code at the end as well, if you are curious and didn't get to scan it. So we have this amazing platform that is true to what we see with the shrimp. What is the first question that we ask? The first question I asked was, shrimp are heavier than water, so they are negatively buoyant. If they are swimming forward, how come they're not swimming forward and going down? What is happening, and how are they creating this force that keeps them at the same height in the water column? This really was, I think, the most interesting question for me, so it was the one I answered first. I hypothesized that during the power stroke, you would have some amount of lift being produced when this angle of attack, and I'm going to use a very aerodynamic term, angle of attack, which is the angle at which this paddle is facing the flow. You would have more lift at this point, less drag, and as the appendage makes its way through the power stroke, your amount of lift would reduce because the force in that direction is less, and your drag-based thrust would increase. That was my hypothesis, and I wanted to understand if this was in fact what was happening. So here I'm showing you a PIV field for the paddle that you see here. So what we're looking at is there's a camera on this side. I have my laser sheet right here, and I'm looking at it from this angle. So when you see the paddle come into plane, that's what's happening here. I do need to give us kind of a note that there's a little bit of clumping in here. The PIV particles are a little bit clumped up. I did not mix my glycerin water mixture for long enough, and once I started the experiments, the glycerin kind of started to separate, but we still don't see that clumping where we're really interested. And we have repeated these experiments a few times because this is a really interesting result. So you see here the raw PIV field. This is our paddle that's going through. You have the velocity and the vorticity here. So what you see is that there's the creation of these two vortices, one here and one here. And we're gonna see the vorticity field. The black is a shadow. I had to paint this paddle. But now you're gonna see there's a shear layer that forms, and then you see the generation of a leading edge and trailing edge vortex on this paddle. This leading edge and trailing edge vortex shed once the angle of attack is too high, and then they become side edge vortices that conclude the power stroke. So here you see the vorticity. Once again, this is the raw field with the particles. Then we're going to look at the velocity. You can start seeing what's happening here. You see this area of vorticity. And once again, we'll look at the vorticity plots so I can perhaps convince you. Hopefully it doesn't take a ton of convincing, but so that you are able to see what is happening. So we thought, insects use this mechanism to fly. So maybe shrimp use this mechanism to generate lift. And as they swim forward, they don't have to make an extra effort to keep level with the water column. So we used the robot for the PIV, and then used the simplified model that was of the endopodite and exopodite only. See, right here, these two, to measure the forces. The pteropodite has been shown to not have a large influence in the generation of forces, and so we neglected that part, simplifying our model to better understand the contribution of these two parts of the appendages. And so I'm showing you a thrust coefficient plot, lift coefficient plot. Here are the magnitudes. These are dimensionless. On this side, you have the angle psi, which is the overall angle of this plate. And here you have the time. So this is throughout a whole power recovery stroke. The most interesting thing, as we saw before, we see the generation of this leading edge vortex. And as we measure the forces, we see that while this vortex is created and sustained, we see an increase in lift. So that's what you see here. And this image is of this exact moment in time. On the endopodite, so now we're moving up to the thrust, and the plane has changed. So now we're imaging from this side. There's a camera on this side. We have a laser sheet right here. And what we're looking at is the generation of a tip and bound vortex, which have been shown to generate thrust. And that's what we see here. And then as we move on in our power stroke, we go back and we see that the leading edge vortex has now shed. So this is the remnants. And now we have this side edge vortex. And once this vortex has shed, you can see that we have minimum lift. Here we see a little bit of a bump in thrust, which I believe is because the leading edge vortex sheds, and now this paddle is contributing to thrust only. And then as you reach the end of your power stroke, your vortices are going to shed, forming a stopping vortex, which means your minimum in thrust. And now we're going to begin the return stroke. Your two paddles are now one on top of the other because they have closed. They begin their return and they generate these two vortices. The plane, as you can see here, has changed from being vertical to being horizontal, and we're imaging from the bottom with the camera. And as this return stroke continues, we see the strengthening of the vorticity, and we see an increase in thrust in our force measurements. This is a very long slide, so questions? Okay, I'll bring candy next time. I'll make a note for that. Yeah, sure.
I guess, how did you decide what laser plane to use on that exopodite? Because it seems to only go 2/3.
Yeah, so we did actually multiple planes.
Okay.
We did 7 planes in total. And then after processing the PIV, we chose the plane that had the best view over the leading edge vortex. But it is sustained throughout the whole exopodite. It's just a different size.
Oh, thank you.
Yes.
It looks like your exopodite is fluttering, maybe because it's passively held on there. It's sort of maybe reacting to vortices that are being shed. I was wondering if you can see the influence of that in your data, and if you think that you would see a different vortex pattern if it was, being controlled more than being passively reactive.
Yeah, sure. So there are a few things that I can speak of for this data, which is at this moment we're not interpolating the kinematics at the correct amount of temporal resolution, and so what you get was kind of this jerky motion that you see that was not sufficient, didn't have enough amplitude to actually shed the vortices. So you continue to have them, but it does make a difference in the kinematics. And yes, there is an influence of the return, the vortices that are shed during the return on the power stroke. The reason why I'm not extremely concerned with this is because my robot uses a flat paddle that is not flexible. Shrimp actually have flexible appendages. So during their return stroke, they're not shedding these side edge vortices that I see in my robot. But yes, they absolutely make a difference. I don't know if I answered your question. Yeah. Any other questions? Okay. Yes.
So shrimp have all the little hairs that must also be contributing to the hydrodynamics here that is not shown in the robot. And so I'm curious what effects those hairs would have if they were added into the model, and what kind of planing do they create?
Sure, that is an excellent question. I do have the answer to that question, but not in this presentation. But I can tell you that the hairs, they are such fine structures. They're actually only 4 micron apart. And because of the boundary layer created around, not a lot of fluid is going through. So they mostly act as a membrane. So what they would do here is that they would increase the amplitude of the forces, but we don't believe that they would change the way the force plots look, just the amplitude. And that's something that we're actively working on, is increasing the complexity. So here we really understand the value of kinematics, and now we want to understand the value of morphology. So adding the hairs and the flexibility will give us a larger picture. But for now, we really wanted to focus on the kinematics. Yes?
So in your final robot, it looks like right now you have just like one side kind of simulated. Will there be 2 sides? Okay.
Yes, there will be two. I started with just one to really understand what was happening in this near field one appendage.
Yeah.
How much information can we take from this, and then we move on. Now we have a full robot, but it is not my work, but if you go on YouTube and look up shrimp robot, you can definitely see it. So I talked about biology-informed engineering, but I also talked about engineering-enabled biology. And this is where it comes in. This is where we understand some of those subtleties. So I'm showing you two different BIV fields. They were recorded one right after the other. The first field that you see here is for krill. Krill are pelagic, so they live in the water column. And they very rarely go to the bottom, from my understanding. And then on this side, we are replicating the kinematics for marsh grass shrimp. Marsh grass shrimp, as you can guess from the name, live in marsh grass. And they don't have to swim as actively as krill, so they mostly are benthic. They live at the bottom of the ocean. That's where they spend most of their time. So if we look at the kinematics, so how things move, I have the plots right here. And in red, you have the kinematics for krill. You can see that their strokes have a larger amplitude here as well. And in blue, I have the kinematics for marsh grass. Their stroke has a smaller amplitude. And what we're gonna see, if you look at both videos, I'm letting them play, here we have krill. And what you'll notice is that this exopodite starts getting out a lot faster. So looking at this is happening at the same time. So both these videos are at the same exact time of your power stroke. And you see that in this one, we have the paddle here, and now it starts going down, whereas this paddle starts, the exopodite extends about here, but not here. And what we'll see here that is super interesting is that krill, with their larger amplitude, allow time for the development of that leading edge vortex that generates lift, whereas marsh grass, due to their kinematics, have a much smaller amount of time for the generation of the leading edge vortex and have much greater amount of time for the side edge vortices. This makes sense because krill need to generate a lot of lift, but marsh grass don't. This was a very interesting result from this study and really shows how we can modulate lift and drag to adapt to the environment by simply changing the kinematics. So my next part will be on exactly this. And if there's questions, please ask them. Credit for the images right there. He has hundreds of these images of shrimp. I was only allowed to put this one in. I had so many others. Next, I want to really explore this ability of shrimp to generate leading edge vortices and understand how that influences their locomotion. We all good? Yes, question?
How did you decide on what species of shrimp that you would focus in on?
With what was available to us in the field. In Rhode Island, we have a lot of marsh grass shrimp. So we did a lot of marsh grass shrimp. Previous research was done on krill, so we used those. We also have some rockpool prawns in the Rhode Island coast, so we got those as well. And actually from here, the Aquaculture Center, we got some Pacific white shrimp. So it's mostly what is available to us and what we can keep in the lab. They have differences in the amplitude, and there might be some minor differences, but they all just use this metachronal wave and modulate the area of their appendages. So you can kind of look at the fundamental principles using one species and then maybe using a robot like we do, understand how that changes throughout. Okay. So first, this work was done in collaboration with Maggie. She's an undergraduate in our lab. She did a lot of the manufacturing for this, a lot of 3D printing with resin, and a lot of troubleshooting. And she was great help for this, so I really need to give her a shout out here. And then we'll move on to this, which was, we saw how big of an influence this angle of attack has on the generation of lift. So what if this angle of attack is not fixed? What if you change it? And we wanted to look at that. And what we did was we swept through the possible values for the angle of attack. So if you were looking at a shrimp that is going at its power stroke, and this is the exopodite here, this is the endopodite. This is a cross-section of what this appendage looks like, and this is at a certain angle. You can vary this angle by varying this zeta value. So what I'm showing here is different decomposition of the forces as you move through this zeta angle. This is at a fixed point in your power stroke. So let's say you're midway through the power stroke. If you vary this zeta angle, what is happening to your paddle? And that number, so the increment angle is right here, and your angle of attack is right here. So as you can see, as you go through different cupping angles, your drag and lift are going to have different components in the two directions. And we wanted to really explore this. And so we designed a little paddle that could allow us to understand this. I have not been able to find a good way to describe this angle to someone who has never seen it and have them understand it the very first time. I think it took my advisor like 6 months to actually understand what was happening here. So if you have questions, please just let me know. But what we're seeing here is that you have your endopodite, which is this yellow line right here. And you have your exopodite. And they are at a bisecting angle. This is what we're calling zeta. If you were to look at our robotic or our little model, this would be your endopodite, and this would be your exopodite. And now I'll show you what that looks like. Watch it again. So what we did was we swept through these angles from 0 to 80. And what I'm showing you here are the curves for this. So we tracked angle gamma for these different angles of zeta. And here we're plotting those values. And you can see the difference that this angle really makes if you compare the 0-degree case, which is at the top, with the 35-degree case, which is at the bottom. The videos are at the same speed, but they are playing at different rates. That's why you see one going faster. And 35 was chosen because this is the angle at which shrimp actuate their appendages. This was the angle that we saw at the beginning. You can see that at 0, there's really no movement of this exopodite during the power and return stroke. But at 35, this side force helps it open and close. We compare this to data for marsh grass shrimp, and we see that there is a similar trend where you have an increase in gamma and then the decrease, and gamma remains mostly closed during the return stroke. We see that here as well. So we see an increase and then decrease, and then it remains mostly closed. One detail here is no one has really been able to understand if shrimp have muscles that pull their exopodite and endopodite, or if this is a passive method. From looking at this data, it looks like it might actually be a little bit of both. Because if you have these fluid structure interactions, you still see the same pattern, but things are really not happening at the same time in the stroke. So we feel like maybe they kickstart this motion, and then the fluid structure interactions help it further along. So, and now one of the really interesting things that's happening in the field is that everyone is trying to create this mathematical model with all the information that we have. Can we create a model that allows us to estimate the lift and drag on these appendages? There's been a few attempts so far, and they've been some successful, some not. And I really thought that we could estimate these forces. So this is a form of Morrison's equation. I am sure it is a very popular equation around here. And I say it's a form because there are a few differences. So I still calculate the Coulomb-Carpenter number, the Reynolds number, and I use Sheehan Buchanan's empirical relation to calculate the drag coefficient based on the Coulomb-Carpenter number and the Reynolds number. The two major differences here are that Morrison's equation is for an oscillating plate. And so what we assumed was a constant velocity profile, whereas we have a rotating plate. And so we accounted for the difference in the velocity from the tip to the root by multiplying the drag by 1/3. There is a velocity integration that gives the 1/3 here that will multiply by 1/2. And then the other thing we did was we added a variable area. From the plots I showed you before, you see that gamma opens and closes, and that changes the effective area, the frontal area, at every step of your stroke. This is the plot for what the area looks like. And so what we did is we made area a function of gamma, your angle at which it's opening, and a function of zeta, which is the angle at which your paddle is going. And then to account for this paddle having different contribution to lift and drag throughout the power stroke as it's moving, we multiply it by this matrix, which has the cosine and the sine, to decompose the perpendicular force with a drag and a lift contribution. And this, for example, if you had an angle of 20, this is what your paddle, the plane that bisects your exopodite, would be doing at every step. So here, the black line is the beginning of the power stroke. It goes through the return stroke, it becomes a little bit lighter gray, and as it comes back, so the colors really are there to illustrate the motion and at what time of the cycle we're looking at. And you decompose the forces based on this angle. And here, for example, this is zeta of 20. Here you have zeta of 60, and you can see how the angle makes a huge difference in the decomposition of your forces. Here, right now, you probably have an angle of attack around 15, and here you actually have a negative angle of attack. So this zeta angle makes a huge difference. And I will show you the results that we got with this model, which I think were really incredible. Here on this side, you have the forces from the model. This is the drag force, this is the lift force. And here on this side, you have the forces from our experiments. So here, once again, we have drag, and here we have lift. The root mean square error is very, very small. So we were very happy that it seems that this modified model can predict both the magnitude as well as the shape of the forces that are produced. And one interesting thing, if we plot the velocity and the acceleration, we see that the force is mostly due to the velocity, so most of the force is due to drag. So I want to end here by kind of recapping what we saw right at the beginning, which was we took this integrated approach of biology-inspired engineering and engineering-enabled biology to understand the fundamental principles of shrimp locomotion, and then understand programmable kinematics that could lead to different forces. So you could have the same robot, and if you have different kinematics, you can adapt to the propulsion method that you want. If you want to go up, you can angle these exopodites in a different angle. It is a really neat way to modulate the forces. And then on the other side, in biology, we understand that shrimp may adapt to their environment by also changing these kinematics. What I have coming up is more of a passion project, but I really wanted to share this with you, is we're trying to understand if we have shrimp at all sorts of scales. And here, this is a Hawaiian shrimp. It is 3 weeks old, and it was 2 millimeters long. Here you have the adult Hawaiian shrimp. All the scale bars are the same, they're 1 millimeter. And then you have a marsh grass shrimp. All these shrimp locomote in the same way. Metachronal swimming, their appendages do the same thing, their appendages have the same morphology, they bend in the same way. So how are they adapting to the lower sizes, lower Reynolds number? This is something that we're trying to find out, and we've done all the PIV videos. Now we need an army of undergrads to track all of the kinematics. So this is really what's coming up. With that, I end my talk here. You could see a Pacific white shrimp, which is the last one in our scaling and was donated by UNH. And if you have any questions, please feel free to ask. Yes.
It's a simple question.
Okay.
I'm just.
Those are the hardest, though.
No, I've been wondering here. You mentioned that the project itself is going to take 10 years, right?
Mm-hmm.
And right now, with the current technology,
Yeah.
what is the size that we can do it?
Great question. I wish you wouldn't have asked, but of course. Yeah, right now it's about this, the whole robot is about this big.
Okay.
It has 10 appendages, so about half a meter. We're working actively with JPL in California to downsize this and use other technologies that are not servos to actuate this mechanism. But it's really going to be something that continues after I leave the lab, and will, I'm sure, continue on for a long time.
Just follow-up question, sorry. So it's half a meter robots.
Yeah.
And if you downsize it, what type of sensor would like to use it? Like what when collect it in the ocean? What can you use it there?
You're asking all the wrong questions, to be honest. So we are planning to integrate a camera, some sort of laser system, temperature and pH meter, perhaps chlorophyll as well. And really depending on the size of this robot. And because it swims in this intermediate regime, we kind of have some leeway on the size that we can make it. But those are the first sensors that we want. We want a camera. We want to measure pH, temperature. But the goal, I think the long-term goal for this project is that we would have multiple of these. you could have a whole swarm of these, and each of them takes up their individual area to map, and then you recover them, and you have your data.
Yeah, that's cool.
So this is a long-term plan, of course, but I feel like we're on the way there by understanding what's happening with shrimp and then applying it.
That's great, thank you.
Yes.
Do you have like an ideal size you wanna get it to eventually?
The goal was to have it real sized, but the real size is 3 centimeters, very small. I am of the opinion that it's actually the right size right now. Because if you make things way smaller, you can disperse them in the ocean and you might not be able to recover them. So if you make something slightly bigger, it still will have all the traits and maneuverability, but you could probably recover it a lot easier. But I am looking into that. Once I move on and do my postdoc, that's actually the question that I'm going to answer, is there an ideal size? Yes. I'll come to you.
Twofold question, but
Should have looked at both arms then.
yeah. The first question is, I'm just curious, the videos are beautiful. How do you bait a shrimp to swim from one end of a tank to another? Because they're not really food motivated. I don't think so.
No, you don't bait them, you wait. You sit and you wait for 6 or 7 hours, and you'll get one video, and then you do the same the next day. And then your hair starts falling out, and your partner never sees you anymore, and so for small shrimp, we wait. We do motivate them. Some of them, maybe we pick them up in a cup, and then we reset the cup and wait for them to swim out. With big shrimp, this is why I love working with these Pacific white. You grab them in your hand, you give them a little flick on the side, and they will swim away beautifully. So if you have to do any sort of PIV with shrimp, this is the one you want to take. Yes.
That kind of leads to the second question, because I'm also used to, like, if you touch a shrimp and it gets scared, it just jets.
Yes.
And I know you didn't really study jet, but I'm curious if you have an opinion, like a thought. When a shrimp jets, is that actually a controlled motion or is it random? Like when it tucks its tail in and bounces away from your hand or
What
whatever?
do you mean as the controlled motion? Do they?
Like, is it sort of a random, I'm trying to escape, or is there actually, like, do you think it's actually like, I wanna go specifically there, and I
No.
wanna hit the wall, or I wanna hit the plant or the other fish over there?
No, I think it's random. We tried that with shrimp. I have some videos, but didn't analyze them, so the PIV doesn't have any vectors, but I can definitely share them with you if you wanna watch. They will just go anywhere. They'll go against the glass, they'll jump out of the tank, which has happened before. They go anywhere, so I don't think they can control it. You can certainly domesticate them, and the more you touch them, the less they will jump. And it gets to the point where you can grab them and they will not jump. So that's kind of what we had to do, was acclimate them to having hands and being grabbed and all these things. Yes.
So you mentioned that shrimp have this eye that can see many more spectrum of colors. Do shrimp have any additional senses, like shark has a sixth sense of electromagnetism? And is that anything that you're looking into environmental sensing?
They definitely have some chemical sensing, because when we drop food pellets, it takes them 30 seconds. You know exactly when they know the food is dropped, because they're very still, and then their arms go crazy and they're just all over the place. You definitely know. So they do have chemical sensing, and we know this in the ocean. They also have chemical sensing. I am not sure about other things. This is totally off the radar. No one heard this, but we think they have little pitot tubes in their appendages, because a lot of their hairs will fold in, but there's 3 or 4 hairs in each appendage that never fold in. They always stick out. So we think they're measuring flow velocity. So that could be a really interesting one, but this was just from a lot of microscopy, so I can't be for sure. Yes.
So I guess this part of the project is modeling lift and drag. Is there another separate, or are you transition towards, I guess, navigation and control in terms of, I guess, do they navigate with a differential between the two PALs, or is it more of an airplane angle flying, I guess, kind of?
We don't know how they turn right now specifically. We think there might be some sort of differential, but we don't know how they turn. That's, so I'm finishing up the PhD, and this is kind of all of the rabbit holes I went down. We have 2 other PhD students in the lab that are looking at that. How do they turn? Can you use just different kinematics to turn? Then we have another section that is trying to make this much smaller and actuated not by servos. So we're kind of working on all fronts. This is the front I decided to take because, yeah, I mean, who doesn't love a rabbit hole? Yeah.
So I know that it's like definitely a future graduate student that would study this, but I'm curious about all 5 of the appendages operating at once and how, like, maybe if you have a hypothesis about the vortices as they're shed from each appendix and like how they might interact given the lag between like the back appendage and then all the way to the first one.
Sure, yeah, we do have the robot that does that, and we have looked at that. It's just because it's not my work, I really am gatekeeping you from it. But if you go on YouTube, you'll be able to see it. What I can say is we've done that work with both shrimp and the robot. One interesting thing during the return stroke is that there is a side edge vortex, like you see, much smaller one, that forms here. And because these appendages are all joined together, this actually never sheds, and it just gets passed on to this one, 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 and then here it goes. So they definitely have methods that pass the vortices along. During the power stroke, we feel like the vortices are well represented in the robot because each one of them does it by themselves. But definitely the vorticity is going to be different when you have more appendages and flexible ones. Yes.
I'm curious too, have you taken a look at buried shrimp? And if so, have you put thought into like, as you're designing a robot, like how you could potentially set it up like a buried shrimp?
Yes. So that's been a great, I have like a million interests. That's one of them. We have done PIV with buried shrimp. I haven't been able to look at it, but it's very interesting. Buried shrimp are shrimp that have eggs in between the legs. Actually, one of the pictures that I have here, you see, is of a buried shrimp. So this shrimp actually has eggs right there. Let me see. So these are shrimp eggs. So this shrimp was buried. It's very interesting because they have this clutch of eggs that in between their legs and they're still swimming. That's a great question that we're thinking about, especially for payload delivery. Because if they can continue to navigate with this huge payload, there must be something that they're doing correctly that we could probably understand. So definitely looking into it. I have a lot of ideas for this, but not enough time. If you're into shrimp, definitely a great research area. Other questions?
I just had one comment online.
Yes.
Thank you for the impressive talk. But otherwise, if we have no questions, let's thank our speaker again.